in terms of multi-factor analysis and our understanding of the organization of brain activity. And also as a side goal is to explain the concept of multifactoriality and how it's estimated to a larger audience of neuroscientists and explain brain criticality to people who are interested in signal processing. So this presentation will first start with a little introduction to the concept of multifactoriality and brain criticality, which will be followed by the presentation of the first contribution that makes up my thesis. Then I'll move on to the second contribution, which will be presented briefly for the sake of time. And then I'll move on to the third one, which is about multi-factor analysis of MEG data. Then finally, I'll do a short presentation of the multi-factor analysis toolbox I've been working on, and then I'll conclude after that. So let's start with the basics. So what we're interested in is analyzing the dynamics of continuous recordings of brain activity. So we have multiple different modalities for this. And so in our case, we're mostly working with MEG and EEG because we need enough temporal resolution to properly sort of follow what the complex brain patterns of our interests are. And in our case, those are brain oscillations which require good temporal resolution, which is what we have here. And we're interested in time-continuous dynamics that are not locked to a specific stimulus, but instead allow us to get a good idea of what the brain is doing on its own over time. So we need to investigate for that the whole recordings. We cannot cut the signal into pieces because there is actually information contained within the length of that recording. And in general, the brain's dynamics contain complex patterns that are not so easy to characterize. So we have oscillations, waves, spontaneous activity. And for that, we need proper analysis tools. So a quick word on magnetoencephalography, which I will be referring to later. So the basic idea is that it records magnetic dipoles coming from the brain, which are generated by neural assemblies within the brain that form unclosed circuit loops. And we can then take those recordings and project them onto the cortical surface. So what is called source reconstruction. And then we can average those reconstructions or parcel-wise over the cortical surface. And so then you get what I described as oscillations, which are these patterns of repetitive activity within a certain frequency band, kind of like so. And this is the sort of thing that we're interested in analyzing and characterizing. So a key feature of brain activity that I'll be working with and is sort of underpinning of this thesis is that brain activity displays scale invariance. So what is scale invariance? In short, it's any kind of relation that follows a power law, which is expressed as a function that scales as the power of its variable. So the reason why it's scale invariant is, well, you can rescale the input variable by a factor, let's say 1 over b, and then you still have at the end a power law scaling with the same scaling exponent, which means that it's invariant. The scaling itself is invariant to rescaling the input variable. And what does it look like? It looks like a linear relationship in log space. So if I take the log of my function and the log of my input variable, I get a linear relationship with the slope that is the power law exponent. So in recordings of MEG, we observe power law scaling using DFA analysis, for instance. And so this has been identified in this seminal paper from 2001 from Lingenka and Hansen. So what you have here on top is MEG signal that then you can integrate. Then it is segmented into different windows of different sizes. And then what you see is the scaling of the variance of the signal in each window after detrending is a power law scaling, because in a log-log plot, you have this nice linear relationship. So we need to be careful here when we select the window sizes so that the analysis is meaningful as well. So now we'll see a set of conditions in another domain where you can also find scale invariance from simple local interaction rules. So I'll be introducing the concept of phase transitions and criticality here. So first of all, we're talking here about complex systems which have large numbers of agents with local interactions. And the sort of key simple example here is the Ising model of ferromagnets, which is described by the Hamiltonian of its representation, where you have spins sigma, I, and j on the lattice, and the neighboring spins interaction represent the cost or not of energy, depending if they are aligned or not. So aligned spins do not cost energy, but then misaligned spin increase the energy cost. And what we can do is we can alter the control parameter, which is here the temperature, and that changes the phase of the model. So for instance, in the 2D and 3D icing model, we have the fact that we can go from a temperature that is below the critical point, where you have a single magnetized state, where all the spins are mostly aligned to the global magnetization. On the other hand, you have a supercritical temperature, where all the spins are misaligned, and that is because basically there is too much energy and it overcomes the local coupling of the spins. And then at the in-between points, interestingly, we have like a different kind of state that emerges, precisely where the transition occurs, where we have different kinds of properties. And those kinds of properties are scale invariance. So the way we identify the scale invariance in this case is by looking at the scaling of the correlation function G of R. So in the icing model, for instance, the G of R would be just the pairwise correlation of spins separated by a distance of R. And so what we can see is that in the different phases, the scaling, this correlation function G of R scales differently. So both in the subcritical and the supercritical cases, there is an exponential term that dominates over long ranges with a characteristic constant of value psi. And that means that basically the correlation between the spins decays very quickly as the distance increases. On the other hand, at the critical point, the exponential term vanishes. And that is understood as the fact that psi diverges to infinity. So the exponential terms then becomes 1, and you are left with a Paolo scaling, which is of the form that we have just seen. And so the feature of scale invariance is that structures emerge at every scale. So here you have like a progressively zoomed in picture of this icing model. So of course, there's the scaling limits of the simulation, but the idea is that the statistics of the model are preserved across multiple different spatial scales. So then, well, we have seen that there is scale invariance in the brain, and we also have seen that critical models and criticality in general expresses scale invariance. So there is a natural sort of idea, which is that, well, then the brain might be also behaving a little bit like a critical system. So the idea is that we consider the brain as a dynamical system, which is operating near a critical point of a phase transition. And the idea is to explain what we observe in terms of scale invariance observed in the brain. So in avalanches, which I won't be describing here, we also find Paolo scaling. And in oscillations, which is what I have shown in the earlier slides from the 2001 figure. So there are advantages that this criticality supposedly brings, and that is supported by modeling, namely that it improves the brain's ability to store the information, transmit the information across different brain areas. Also, it improves its computational power. And the way that the brain regulates this is by homeostatic regulation, which in a healthy human naturally adjusts the sort of critical point or the control parameter, the sort of control parameter of the brain close to the critical point so that it behaves optimally. So how do we characterize scale invariance in brain time series? And in recordings of brain activity, we have 3 main approaches. So in the brain criticality literature, we often like to look at the oscillations present in the time series, which will appear as peaks in the power spectrum. And by doing some analysis to extract the envelope of those oscillations, so basically the envelope of the time series bandpass filtered around these specific frequencies, we can see that those are scale invariants expressed as this linear relationship in the log-log power plot. And so that's what the brain criticality literature usually focuses on. We can also use avalanche analysis, as mentioned. And finally, we can also look at the low frequency fluctuations of broadband recordings of brain activity. So in neuroimaging, we often find that the low frequencies also scale as a linear relationship in this log-log plot. And that is where we can apply our scale invariance analysis again. So in practice, we use estimators to derive the scaling exponents of the time series, both in the low frequency regime here and in the envelope scaling. So there is the power spectral density, which is not the very good estimator because it has some properties that make it not reliable for certain classes of time series. Then we have the degenerate fluctuation analysis, which is more popular in the brain criticality literature. And finally, we have the wavelet Bayes formalism, which is more common with people who work with multifactorial analysis. So I'll just do a quick brief on what is fractal and monofractal and multifractal time series and the notion of pointwise regularity. So if we look at a monofractal time series, we can draw the associated multifractal spectrum, which determines for each for all of the time series what is the pointwise regularity of the time series. And by pointwise regularity here, what we mean is the degree of smoothness of the signal. And so you can imagine that a regularity of 1 is exactly a continuous signal. A regularity of 2 is a derivable signal with a continuous derivative, and so on. And so in between, we also have actually classes of signals which take values in between discrete values. So in this case, for instance, we have like a random walk that has regularity 1.7. And so what this spectrum represents is that in this monofractal time series, every time point has the same regularity. So we can then play with the regularity a little bit to see what happens. So if I decrease the regularity, I get a rougher and rougher time series until I get something that looks more like a noise. And if I increase instead the pointwise regularity, so that all the points have regularity 2, here I have a very smooth time series, which is actually derivable. And then that's just a very basic concept. But then what we do is we introduce the fact that there can be more than one pointwise regularity in a time series. So in scaling invariant time series, sometimes we have what we call a multifractal time series. And those multifractal time series, they have this special property that there is not just one pointwise regularity present in the time series, but there's actually a whole spectrum. And so you have points with different local pointwise regularities. So for instance, you have points where there are jumps, which corresponds to low regularity locally, and points that are very flat and smooth, which correspond to very high regularity locally. And what's interesting is because this signal is scale invariant, if you zoom in, you still have the same distribution of jumps and flats. So even if I was going to zoom on the flat point of my time series, I would still find some jumps relative to the temporal scale I'm looking at. So by adjusting the width of my multifractal spectrum, I control how widely the regularity fluctuates within my time series. And so if I look at the brain time series that has been integrated, for instance, you can see that it has a multifractal spectrum that looks more like a multifractal time series than a monofractal time series. So in fact, we see in practice that there are some brain time series which show multifractality. So how do we estimate actually the multifractal spectrum in practice? We can't exactly go and look at the pointwise regularity of each time point of our sample time series. That's just not feasible. We would need infinite sampling frequency for this. So instead, we rely on multi-resolution quantities to characterize the scale invariance. So like a natural example of multi-resolution quantity would be the discrete wavelet decomposition, because it naturally decomposes the time series in a non-redundant decomposition, and also it decomposes the signal into sort of temporal shifts on the index here, k, and then temporal scale on the index on the y-axis, j. And the scales increase logarithmically, which matches what we are using for estimating the scale invariance. So the notion of pointwise regularity is fundamentally related to the multi-resolution quantities that we choose. And there is this nice theorem that says that basically the choice of multi-resolution quantity informs the definition of regularity that we're working with. So for our practical analysis case, we use the wavelet p-leaders because the wavelet coefficients are not suited for multifactorial analysis. They have inconvenient properties. So instead we rely on the p-leaders, noted LP of JK, and they correspond to a different notion of regularity than the one I introduced. It's called the p-exponent notion of regularity. And they are obtained via computing a weighted p-norm over the set of wavelet coefficients. So here we have the wavelet coefficient decomposition of a time series. And to compute a p-leader, I would have to, if I want to obtain this p-leader, I would have to compute the weighted p-norm over this whole set of wavelet coefficients. And what this does is it stabilizes the estimates, and it allows us to get good and reliable estimates of the multifractal spectrum. So we can't exactly just use the multi-resolution quantities by themselves. We have to compute some scaling functions from which we can estimate the exponents. And then what we're doing is we are characterizing the multivector spectrum by the log cumulants. And so we basically, what we like to have is a few scalar quantities that describe the properties of the multivector spectrum. So the log cumulants, the CM, they're very useful for that because they describe both the mode of the spectrum, its width around the mode, and for instance, the asymmetry if you go into even higher orders. And they operationalize concepts of scale invariance. So in this case of C1, it's operationalizing the concept of self-similarity. So how does the signal scale as a whole when I rescale its temporal scale? And then C2 operations is multifactoriality. So how much fluctuation there is point-wise of the regularity of the time series. And so to estimate those log cumulants, we actually can just use the logarithm of the p leaders. And under the assumption that our time series is scale invariant, then this forms a linear relation between the temporal scale and the function Cm of j. And so we estimate the log cumulants by just using linear regression over the functions Cm of j. And so those gives us the properties of the multivector spectrum in a few convenient values. So there is some prior out on multivector analysis of neuroimaging data. So it started used on fMRI with the investigation first of just resting state data, but then soon enough people started applying it on task conditions as well. But the thing is that's a low temporal resolution Bowl signal, that is the blood oxygen level dependent response. And it's not what we're looking for here because we're trying to estimate something that is related to brain criticality, and that's too low temporal resolution for us. So there has been also work done on the low frequency fluctuations in EEG and MEG, and on the envelope of the oscillations themselves, which is what the gold standard for brain criticality analysis. There has been some contributions on epileptic seizure identification and brain-heart interplay that have involved multivector analysis, but in both those cases, and there's some more that have cited in my thesis manuscript, they only use multivector analysis as a tool to give an estimate, but they don't seek to interpret what does it mean. So our goal here is to say, well, we have this multivector analysis, which is just this nice and fancy estimates of multifactoriality, but what does it mean in terms of what the brain is doing? We are looking for a way to interpret the results of multivector analysis in terms of sort of brain dynamics and the properties of the brain. So the first contribution towards that was this manuscript, Multifactoriality in Critical Neural Field Dynamics, which I have worked on first. And the idea was to simulate a model that comes from brain criticality. So the brain criticality hypothesis states that, well, the scale invariance comes from the critical nature of the brain. So we have this multivector analysis that fully characterizes the scale invariance of the time series. So that feels natural to apply it to those critical systems. But we don't have, up to that point, we didn't have like a good modeling basis for just justifying applying multivector analysis to critical systems. There was no model which was demonstrated to have multifactoriality. There was also a model for brain criticality. So we use this model, the Landau-Ginzburg formulation of the Wilson-Cowan field equations. And it's a system of stochastic differential equations with two fields. So the top equation represents the excitatory activity field. So that's the fast dynamics. And it's represented by this blue lattice here. So we also have below the inhibitory activity field. That's modeled here as resources activity field, but in effect, it is inhibiting the activity of the excitatory field. And those represents the slow dynamics in this case. So we can play with the parameter tau d here, which we picked as our control parameter, to have the model undergo a phase transition all the way from below criticality, where the model displays intermittent activity that you can see here below, all the way to stable oscillations for values of the control parameter above the critical point. And what's interesting is at the critical point, we have interesting dynamics where it switches between what's it called? Stable oscillations that widely vary in amplitude, and intermittent activity as well. And there is an interesting fact that we have bistable dynamics slightly below the critical point as well. So it's extended range of the control parameter where the dynamics are bistable, and that will be relevant later. So to simulate the model, we had to use GPU acceleration. And the reason is, well, to do our scaling variance analysis, we need very long time series because we're estimating scaling exponents from logarithmically increasing temporal scales. So we need a very large number of oscillation cycles in this case. And we used a GPU implementation with a custom CUDA kernel, and that reduced the simulation time for around 6 hours for a long simulation, which was reasonable. And we can apply it to other stochastic differential equations as well. So this is kind of example what the model's activity looks like in different regimes. So this would be the supercritical regime, where you can see that the local coupling sort of vanishes almost, and it becomes constant activity. And at the bottom, there is more intermittent activity, and in between, there is this critical state which has interesting properties. So the first thing that we need to do is make sure that the simulation of this model were in fact scale invariant, and we could meaningfully apply our multifactorial analysis. So we need to check that the CM functions that I introduced are scale invariant, and we analyzed both the low frequency fluctuations of this model and the envelope of the oscillations that this model is simulating. And in both cases, over the range of temporal scales over which we are doing the estimation, the relationship is linear enough that we can justify applying multifactorial analysis. And so here we can see that for 2D equals 99, which is around the critical point. We have a nice linear scaling here and here, which is going to be the basis for which we can estimate the log cumulants later on. So by estimating the log cumulants for different values of the control parameter 2D, we can see that at the critical point, where 2D takes its critical value, there is a peak in both the C1 and the C2 metrics. So that means that the self-similarity and the multifractality of the time series is maximal at the critical point of the model. And that is the case for both the low frequency fluctuations of the model and the envelope of the oscillations of the model. And so another important fact is that there is this bistable region that I mentioned before. And in this bistable region, in general, the multifractality is higher than in the rest of the domain for the control parameter. And that suggests that the bistability also is a factor in determining the fact that there is multifractality in the model. So then the next question that we're asking is how important is this property of multifractality? Is it just a trivial property of the model, of any kind of phase transition always will be multifractal, or is it kind of specific to certain classes of models? And so we changed the parameters of the model to simulate a different kind of phase transition. We reduce the level of noise and increase the driving of the model. And then here we obtained a different kind of phase transition where moving the control parameter xi increased C1 at its critical points. So you have a nice peak of C1, which indicates the critical points. But the C2 parameter that we estimate sort of does not show a marked phase transition there. So it's constantly slightly multifractal, which led us to conclude that multifractality was not a trivial property of any kind of phase transition in a dynamical system, but it's specific to certain classes of dynamical systems. So overall, this contribution is first study of the multifractal properties of neural equation dynamics, and especially in the context of brain criticality. So this is showing that there is multifractality in the average activity of simulated neural mass equations. And we have sort of a prediction that we can make that there will be joint variation of C1 and C2, which will peak together close to the critical point of the system. So then there is the second contribution, which I will be sort of passing over quite quickly. But the gist of the idea of that contribution was that this multifractal analysis that we're using, it's quite sensitive to outliers. So even if we introduce a little bit of noise, a small perturbation, and here I mean small in terms of only a small segment of my signal is perturbed, that's enough to significantly affect the multifactorial estimates. So even if 3% of the duration of my time series is affected by some kind of noise source, that's enough to wildly modify the results that I obtain. And you can see here for 2 different time series, simulation here with and without noise. And the clean time series and the noisy time series have this very close values of C1, which means that the self-similarity analysis is not so sensitive to the outliers. But on the other hand, the clean and noisy signals have very different C2 estimates, which means that our estimate of multifactoriality here is deeply perturbed. And the intuitive reason why is because the impact of outliers on estimating higher order cumulants grows as the power of the cumulant order. And those kind of impulsive noise sources, they can be quite prevalent in intracranial EEG, but sometimes also MEG can be quite noisy as well. So I'll just skip over a little bit. I'll just present here the application to a single time series. So here we have a single MEG time series. And the original estimation without any kind of outlier detection gives a spectrum that is deficient because it's not a well-formed multifactorial spectrum, which would lead us to conclude that the time series is not multifactorial. But what we can show is that by applying the proposed method, we detect the segments outlined in blue here as outliers and carry out the analysis on the rest of the time series, and we obtain the results in orange, which show that aside from those few segments, the time series is indeed multifactorial with a scaling exponent that is sort of in line with what we expect from the brain. So now I'll be presenting the third contribution, which is the characterization of multifactoriality in oscillatory dynamics. So here we were working with MEG data, and the idea was to investigate what does multifactoriality tell? What does it show? Is there even any multifactoriality in the dynamics of brain oscillations? So we have established, and there has been, well, the literature is rich in papers showing that there is long-range temporal correlation, so high values of C1 present within the oscillation dynamics of oscillations. And now we're trying to determine whether or not this has anything to do with the multifactoriality of the brain time series. So we're using MEG measurements, and we are using an operationalization of oscillations coming from brain criticality research, which is quite an important detail because other contributions looking at multifactoriality in the dynamics of oscillations were using different ways to characterize brain oscillations, which meant that the results were not directly interpretable with respect to the rest of the brain criticality literature. So our second aim was to provide a standard way to estimate multifactorial properties of the dynamics of brain oscillations. And also we were trying to see if we could replicate the results that we had in the simulations. So our data set is 15 minutes resting state recordings collected from 75 healthy subjects, which the data was then pre-processed and projected onto the cortical surface, and using parcel-wise averaging, which meant that we had time series with 200 parcels. So how we do the analysis? Well, we first need to extract the oscillation amplitude time series. And so for that, we use the complex Morley wavelet filtering between central frequencies around 5 to 80 Hertz. And when we take the absolute value of the Morley wavelet filter time series, we get the envelope of those oscillations. So here we have the raw time series, and here we have an example where we take the envelope. And here we can see already that rescaling this envelope time series has some scale invariant properties because there is a distribution of more and less regular time points. And that distribution seems to be somewhat preserved between the temporal scales. So then after we have extracted those oscillation time series, we compute the wavelet 2-liter representation of those time series. So first we integrate the time series, which is required in the case of wavelet analysis, in this case, because the time series are too irregular to be analyzed without this integration. And also we are choosing the 2 liters because this enables us to compare the results with DFA-based analysis. And the reason for that is the DFA is actually measuring the notion of regularity that DFA is measuring is the same one that the wavelet 2-leader analysis is measuring. And so then from that, we can obtain estimates of the multifactorial spectrum. So here for both time series, we can see that we can estimate this multifactorial spectrum, but more importantly, in our case, we can estimate the C1 and C2 values from those wavelet 2-leaders. So one key thing that we had to do here, and that sort of also sets the stage for the results to come, is we use surrogate analysis to determine whether the multifactoriality that we measure is genuine. So we use two surrogate generation methods. So first one would be to shuffle the phase of the Fourier representation of the time series, which has the effect of preserving the power spectrum of the time series, but it destroys the other properties, like nonlinear properties of the time series. And we also use another method for surrogate generation, which was shuffling the modulus of the increments. So in this case, of the signal before integration. And what this does is also preserves the power spectrum of the time series, but it does destroy the multifactorial properties. So here you have an example of an original time series in blue and the surrogates in green and red. And you can see that they have the same power spectra, but actually quite different multifactorial spectra. And so this surrogate procedure, it does reduce the degree of multifactoriality, which means that what we can do is then carry out significant tests. So first at the group level, we can compare the C2 values that we obtain for parcels at different frequency bands with the average surrogate multifactorial values. So then our null hypothesis would be that the C2 value for the parcel is greater than the surrogate C2 value, which means that if the observed C2 is lower, then it is it's significantly more multifactorial. And at the single subject level, we can also say that looking at the distribution of repeated surrogate estimates, looking at the distribution of estimates from repeated surrogate time series, we can determine that the time series is significantly multifactorial if the observed value of C2 is lower than the 5th percentile of the surrogate distribution. And the reason why we're picking the 5th percentile in this case is because lower values of C2 indicate more multifactoriality. So in that case, lower value would mean significantly multifactorial. So our first result is that multifactoriality is prevalent in both alpha and beta bands. So by looking at the group level statistics, we can see that both for the modulus and the phase surrogate generation methods, the fraction of parcels that have significant multifactoriality for different frequencies, we can see that there is a drop-off after around 30 to 40 hertz. But below that, in terms of frequency, we have a large number of significant parcels. And we can also see that the modulus surrogate method seems to determine that more time series are multifactorial than the Fourier surrogate time series. So it is sort of the Fourier phase surrogates are more conservative than the modulus shuffling. And so one other thing we can do is look at the spatial correlations to determine if there's kind of a difference in the nature of the multifactoriality between the different frequency bands. And so here we have the correlation matrix, where we calculate the Spearman correlation coefficient across parcels of significance. And we can identify distinct alpha and beta band spatial patterns, which we visually identify and separate as alpha and beta bands correlation clusters. So then what we can do is say, okay, well, we're interested in how those clusters are distributed on the brain and what kind of multifactorial properties they have. So in the alpha bands, what we observe is that the median C2, so the median value of C2 across all subjects, is higher in the occipital lobe and also in the parietal lobe compared to the frontal lobe. And we have also, quite interestingly, the sensory motor cortex appearing quite multifactorial. And the trends in the beta band are less pronounced. So the peak multifactoriality in the beta band, it's in the parietal lobe. And in general, we have higher values of C2, which means that in the beta band, the time series appear less multifactorial. And so we had previous findings showing a gradient in low frequency fluctuation analysis. So here we are looking at the envelope of oscillations, but it's interesting to see that there is a gradient present still there. So then we can look whether or not this pattern that we observed in the group level statistics holds at the single subject level. So here, the significance of recordings is determined for each recording. We determine whether the multifactoriality that we observe there is significant or not. And so here again, we can look at the fraction of significant recordings for each frequency band and see that there is a peak in between 8 to 25 hertz. And looking at the correlation patterns, they reproduce also the same correlation patterns. And here we can see actually that the modulus surrogate method seems to have an issue in the gamma bands. And if we look at the time series in the gamma band, actually they have quite a few outliers. So we could not apply our outlier detection method here because it requires removing segments from the time series. But the surrogate generation method, they absolutely require that the time series doesn't have its segments removed because it considers the time series as a whole. So that's something we might work on later. But anyway, the phase surrogate method doesn't seem to be perturbed by the outliers in the gamma band and shows like a consistent spatial correlation pattern. And if we look at the spatial distribution of the fraction of significant recordings, we can see that it reproduces the same pattern that we observed for the median C2 values. And what this means is that basically parcels that are more multifactorial are also more significant, which is kind of in line with what we expect from how the significance test works. So there is one last thing that we looked at, and it's quite important here because it determines a significant part of our multifactoriality estimates. So we observed a strong correlation of the C1 exponent and the C2 exponent across parcels. So here we have, for a single recording, across all the parcels, so all 200 parcels, we observe strong negative correlation of C2 and C1, which means that, well, when there is more multifactoriality, there is also more cell similarity, which is in line with what the model described. And this pattern is also stronger at the level of single recording compared to the whole set of recordings. And it might seem nice that we confirm the result of our model simulations like this, but it does maybe hint to something else. And so to have confidence that this is a genuine pattern, we also looked at the linear decomposition, spatial decomposition of the time series. So if our data is considered as a matrix of size n samples, n parcels, then doing the spatial decomposition of this time series yields us a set of n parcels principal components, where each principal component is a time series. So the first principal component is a time series which explains the most variance. And here we are representing the loading on the first component. And the loading on the component of a time series means basically the correlation between time series. So here, for each parcel time series, on the y-axis, we are showing how correlated that time series is to the first principal component. And so we can see here that there is an even stronger negative correlation between the estimate of multifactoriality and the loading on that first principal component, which is quite strange because, well, if all parcels are independent estimates, there shouldn't be such a strong dependence on the correlation with the strongest principal components of our multifactoriality estimate. It seems to suggest here that multifactoriality is caused by correlation to a global activity pattern, in fact. So we investigated a little bit further. So we looked at the global correlation between the correlation with the first principal component and the C1 and C2 estimates, and observed that there appears to be a strong correlation pattern that is consistent. And we can see that when there is a high loading on the first principal component, there is automatically as well a high value of C1 that is estimated, and also in general a low value of C2 that is estimated as well. And so that seems to suggest that the correlation of the time series to the first principal component underpins finding extremal C1 and C2 values. And then we can go even further and remember that we observed that C1 and C2 are correlated. And in fact, it appears that this correlation between C1 and C2 is in fact mediated by the common correlation to this first principal component. So here, if we look at on the x-axis, we are looking at the correlation between C1 and C2 for each time series, and for each parcel. And here we have the correlation between the loading on the first principal component and the C2 estimate. And so we see that the correlation between the loading on the first component and C2 appears to be a large factor in determining whether or not C1 and C2 will be correlated. So that means that if there is no correlation between the C2 estimates and the loading on the first component, then there is also not going to be any correlation between C1 and C2. On the other hand, if there is a strong correlation between the C2 estimates and the loading on the first principal component, then there is also automatically going to be, at least in the data, what we observe is that there is also at the same time a strong correlation between C1 and C2. So this kind of puts into question our approach for analyzing. So this contribution provided the first characterization of the multifactorial properties of brain oscillations in MEG, and it used surrogates to identify significant multifactoriality in alpha and beta bands. And we show that the variance in C2 appears to be driven by the global correlation of time series to a common leading principal component. So I'll just introduce really quickly. I've been using this toolbox, which I've been maintaining during my PhD to get those results. And it's open source, documented, everyone can use it. And it's meant to be quite simple. So during this PhD, I have worked on producing 3 article manuscripts. So 2 of them I have presented, and the third one is this outlier detection manuscript, which I have briefly introduced. I have also co-authored a book chapter on using weak scaling exponents to estimate the multifactorial properties of neurological recordings, and I have worked on here pre-processing and applying multiple tenses on the MEG data specifically. And I have also made some conference contributions. So in conclusion, this thesis project aimed to clarify the presence of any link between multifactorial analysis and brain criticality in general. So we have identified multifactoriality in the model for brain criticality. We have also, so that should be usable as a starting point for finding more general principle of multifactoriality in criticality. We have proposed an improved method for multifactorial of noisy intracranial recordings, so useful for real-world conditions. And we have characterized the multifactorial properties of brain oscillations and shown that it's quite prevalent and it's associated with the key construct of brain criticality in the form of self-similarity. So for the perspectives, we can identify a few directions where this might then lead. So clearly, we have a need to orthogonalize the multifactorial processes present within the recording. So currently, the data we analyze appears to be a mixture of scale-free processes. So there's currently a master internship project with Sabine, who is trying to integrate new processing steps to improve the orthogonality of those time series. And there is also maybe the possibility to develop a multifactorial analysis-based version of ICA to separate scale-free time series. So we could also look towards deriving analytically the multifactorial exponents from the equations of neural models. So for instance, there could be developments towards building a multifactorial Kuramoto model, or even take other models from physics and try to see, basically this would mean looking at the scaling of different moments of the model, which is quite tricky to do, but it's also quite interesting. And finally, we could also improve in general the methods that we're using for multifactorial analysis. So there is a recently proposed box leader that solves some of the issues that we see. And also there, we could use a stationary wavelet transform or the continuous wavelet transform instead of the one we're currently using to also improve our results. So I would like to thank my supervisors, Philip and Matthias, for guiding me during this PhD and for providing support. I'd like to thank some colleagues and collaborators. So Sheng, who welcomed me to Helsinki University. Then some local collaborators in Helsinki who I have worked with, but unfortunately didn't get to co-author any publications. Maybe yet, maybe they will come in the future. I would also like to thank the members of the Pava Lab who worked on collecting and pre-processing the MEG data, as this was critical for me being able to use it for my analysis. And finally, I would also like to thank the French collaborators with whom I have worked on weak scaling exponents and multi-factor analysis, more theoretical developments. So I would like to thank you for your attention. So I've also included some references for when I show the slides, if you're interested.
Thank you very much. So now we will move to the question and answers. And, can start with Professor Anna Levina. Can you hear me?
A bit worse than Merlin before, because you're standing further away from a computer and I see not your microphone but the computer.
Okay. It's better here now?
Yes.
Okay. So I will leave you the floor for about, I would say 15 minutes. Mm-hmm. Okay?
Yeah. Thank you, Merlin, for this very beautiful presentation and nice video visualizations that you provided along the way. They were very impressive. So one thing that keeps bothering me with respect to considering the brain criticality and classical, by now, detrended fluctuation analysis, that's basically underlying assumes that there is a single Hurst exponent that it would recover. What does, first of all, if the data is truly multifractal, what is the error people are doing while extracting just a single detrended fluctuation exponent? Is it still informative? Can it be kind of tainted because of the multiple exponents that are there? Let's start with this one.
Okay, yeah. Thank you for the great question. Yeah, it's true that the past literature has focused a lot on DFA analysis, almost exclusively. And the one problem with that is, of course, that if your time series is multifractal and you're only estimating a single exponent, then you're missing out on all of the rest of the information that the signal can provide. So in practical terms, there's nothing exactly wrong with using DFA. It's just incomplete with regards to what kind of information that you're describing. So if you think, maybe I can show back the example here. Oh, sorry. At the beginning, I showed the example where you have two time series that have quite different. So here, if you look at this again, you can see that both time series would have the same DFA exponents, but of course the actual properties are quite different. So only relying on DFA means that you have this incomplete picture. So in some cases, it might be actually really relevant to what you're trying to figure out, especially if you have intermittence in the models or any kind of regularity fluctuations that would not be captured at all by the Hurst exponents or the DFA exponents only. So this is why we like to bring multiple analysis into the picture, because it is more informative. There is one caveat that, of course, the estimation errors of the higher order properties of the multifractal spectrums are higher. So when we do this multifractal analysis, we have to be more careful about what kind of data we're taking, because it's more easily corrupted by noise.
So if you take like for the data that you used for your multifractality analysis in the last project, I missed, was there also a picture where you did the DFA on the same data? Did you show that you say it's C1, yeah?
Yes.
That the C1 exponent is the same as the beta of the DFA?
No, I don't have, so I didn't put it in the paper. I did this analysis actually for the MEG data. So from a theoretical perspective, they measure the same concept, but of course it's two different ways of measuring the same concept. So when I looked at the analysis, they have like it's a very strong correlation between the two estimates. So it's not perfect, but it's basically measuring the same thing. I didn't show it because yeah, it's almost trivial in a way, but I guess it's still an important point to make.
And could it be, say, like if you would look at it in the terms of the DFAs, it is for me a little bit easier to imagine, and that you have just multiple slopes in the DFA, like a knick somewhere.
Yes,
would it also induce the perception of the multifractality in there, and how this would relate?
So what do you mean exactly by multiple slopes?
Like if you would go back to the, for example, like the first slide where you introduce a DFA.
Okay. Yes.
You see here, you already like there are two natural cutoffs because of the sampling windows and the noisiness, if I recall correctly, and the largest window you can sample. But otherwise, like if you there is a single slope. You could also not do the cutoff, for example, in the beginning, and then fit it with two slopes.
Yes.
Would the same data then feel like multifractal? And there could be another knick because the process is just not having the same scaling over different size scales.
Yeah, so it's an important point. So it's important to clarify here. This would be a different concept. So I have seen it referred to in the literature as a multifractal crossover, where the dynamics crosses over into a multifractal regime for sorry, a different fractal regime over a certain temporal scale. But in this case, it would be like the notion of multifractality would be if you look at different moments of the time series, the slope, the measured slope on the same range of temporal scales would be different. Would be scaling differently as a function of the moment.
Okay, so a bit different, maybe somewhat related question. So if you would have a monofractal time series, how can you like do you know how can you add a noise to it such that it looks multifractal? Like which types of like the data surrogate models are there, and is it possible to just like fake the multifractality by having some like the intermittent noise you looked at to remove like for the artifacts, but it could also change the scale that you perceive from being monofractal to multifractal.
Yeah, that's true. So in general, for generating multifractal noise, what we do is we use multiplicative processes. So for instance, the kind of one of the first instances of multifractal processes was multifractal cascades, and those can be generated in sort of multiple ways. But it's also true that our surrogate analysis methods, they don't have any strong grounding in the multifractal analysis literature. Actually, there is no established proper way to do the surrogate analysis that is grounded in theory using surrogates. So we took the best thing out there, the most used ones, and in practice, they seem to be like capturing something. Of course, they also have some issues, and we have identified, at least in the gamma band, that it doesn't appear to work as well as we would hope for. But in general, I think there is still some development needed in the surrogate procedure for multifractality. But it's kind of an open question still. And I guess, yeah, this was not the focus of this PhD.
So is there a kind of perfect, like the generative process that would, for given C1 and C2, generate you a process that has these parameters?
So for instance, there is the multifactorial random walk, which is what I use when I did simulations, and that you can obtain by taking a Gaussian noise and multiplying it with creating intermittence by multiplying Gaussian noises. Yeah, I have described it, I think, in the.
Okay, I'll just trust you on that. I'm not a particular multifractality specialist, as you might guess. So when you were doing the shuffled surrogates of the original data, you use the data to data. Like if you would generate the true multifractal process with the C1 and C2 as in the data, would the I feel that then potentially the shuffling procedure would give you a very strong confidence, but like stronger confidence bounds, and if you just use always the data that might not perfectly be multifractal, you just fitted it and estimate the multifractality, which always is possible. Did you try that, or maybe I missed it? You actually did exactly this procedure.
So the surrogate procedure we use doesn't estimate multifractality at all. It relies completely on either Fourier transform first and then shuffling, or just shuffling the modulus of the time series. But so it is not affected by our estimation of the multifractal.
Sorry, I would interrupt a little bit. Like, sure, it is not affected, but whether you are estimating correct multifractality or not is basically a question. And checking this procedure on the surrogate data that is the multifractal data would give you the strong confidence bounds that are where you don't need to check the hypothesis whether it was a multifractal at all to start with.
Okay, yeah, I think that's interesting. We know we haven't tried that, but that's definitely like a good idea, and yeah, I'd be interested in trying that out. I don't have like any particular idea what the results might be just now, but yeah, I think it would be worth trying.
So I'm not sure whether I did not yet use already my 15 minutes. I still have some questions, but we could they're not urgent. We could also do a round and then decide whether there are still some questions left.
There's still some time, so.
Okay. Okay. So for the model, for the measuring multifractality on the Landau-Ginzburg model, it seems like you showed that the transition to and away from bistability are the important points. Are like if you would just take another model that has just like a simple generation, which would have like hysteresis transition to bistability, would it produce the same picture in a time series? Do you know?
So we haven't tried it, but my guess is it wouldn't, because one of the key components of criticality is that it's present in scale invariant models. So that requires some enough, at least spatial or temporal degrees of freedom for there to be this scale invariant relationship. So in this case of this model that we're simulating, it's actually a key component is the fact that it's spatially distributed, and there are like different regions are doing something different. So in mean field approximation, I don't think that we will observe the same dynamics. Of course, we haven't tried it, so I guess we could also have a look. I know that you can obtain multifractality from bistable processes like you described, but I don't know if it would be concomitant with high self-similarity as well.
Yeah, I was just curious because it feels like the transitions are very clearly related, maybe even not to the critical point, but the both transition into and away from bistable regime seems to change your C1 and C2 components. So it might be more related to the bistability than to criticality.
Okay. Yeah, no, that's a possibility, I think. But in that case, we would have to also look at simple sort of bistable models. We also included just a reference at the bottom of this slide that sort of specifically talks about what the properties of this kind of models are in terms of showing both bistability and criticality. So it would be interesting, yes, to see if just removing the criticality aspect and keeping the bistability still shows the same behavior or not. So that would be like a natural next step, please.
Good. The kind of a little bit maybe more speculative question. There are multiple people suggesting that there is a hierarchy of timescales in the brain that somehow brings the idea that it's not everywhere same critical, because some brain regions are dominated by the fast timescale processes that should not actually lead to very long-range temporal, highly perceivable long-range temporal correlations in the absence of stimuli. So do you see any signatures? Like, I've seen your maps, but I had a hard time interpreting them this way. So just like a different way of using the data. But on a global scale, what do you think about this idea that criticality is not expressed equally everywhere? Do you see any support for it in your data, and how potentially the change of the intrinsic timescales can be seen in your way of looking at the data?
So the modeling approach, it shows only, it's a very basic model. So it's only simulating one oscillation frequency. So in that scenario, what we observe is not, I mean, of course, the fast dynamics determine the precise evolution of the model, but it's actually the coupling to the slow varying inhibitory field that enables us to have this long-range temporal correlations. So in the case of how the brain is actually working, I wouldn't be able to tell you. What we have found is that, yes, there is not this multifractality everywhere present in the data that we have analyzed. So within that context, I would say, yeah, it's not guaranteed that the brain is critical in every respect. But as well, I would say, based on how much I have been reading about critical dynamics and dynamical critical systems, there seems to be many ways that dynamical systems can remain critical even with modifications or departures from the usual frameworks, much more so than for static models. So I wouldn't say it's not possible either, that there is some form of criticality present in there.
Like present everywhere? Well,
there could be, like, I guess it depends how you measure criticality. If you do like a global brain analysis and try to derive a single criticality parameter for the brain, that might not be accurate. But if you try to isolate individual components and say, well, alpha band oscillations on their own, they appear to be critical, then it might be like a better first step. And well, if the alpha band oscillations are also affected by other bands, then those other bands are somehow involved in making the alpha band critical. But it doesn't discount the fact that we would observe, for instance, long-range temporal correlation in alpha band or beta band. I don't know if that makes sense.
So the analysis with the hierarchy of timescales would not first filter some oscillations, and it would just look at the autocorrelation function and its decay. And if there is oscillation, you would need to remove the oscillation, or like have a model that explains oscillation and decay of autocorrelation. And this would, like, basically, if you would just have negative oscillation, it would mean that the oscillation would be damped, and the damping of the oscillation would give you the time scale. And it looks like in very different ways of recording the data, including MEG, there is this hierarchical relationship that is more kind of closer to sensory areas have fast timescales, and further away from sensory areas have a slower timescales, which kind of feels like, oh, we would expect them to be more critical. But it is not filtered in its particular frequency bands.
Okay.
So
Yeah.
there might be some interesting relationships, and they might be very non-trivial. With respect to filtering, how, like, when you showed some picture from the data in the very beginning and showed your alpha and beta peaks, not like in the very, very beginning. I don't remember which.
Okay, yeah, in the introduction, yes.
Yes. So I assume this is how the real data look like.
Yes, that's real data.
So if, I don't know, like more mathematics people would think about the peaks, they would think about something that is much more pointy and easy to detect than this one. However, I understand that in real data, it is seldomly that perfect peak that is very easy to locate. How much you actually depend on how broad and how precise you would filter the data before computing the envelope, and what happens if you mix together the real oscillations and non-oscillatory components that are just nearby, and you just mix them because you were taking too broad part of the spectrum.
Okay, so I think the people are making me sign that's the last question I will answer for now. Yeah. So with regards to the appearance of the plots, it's just you have to remember that it's a logarithmic y-axis scale in order to fit the different temporal scales. So it looks a bit flatter than it would if you had the linear y-axis, and that would look a lot more like an actual peak. But then on the other hand, yeah, it's true that we filter the data at many different frequency bands, and we're not guaranteed to have oscillatory components there at all. So this is kind of a debate I have had many times, and this is kind of a standard procedure in brain criticality analysis. So I carried out the multi-factor analysis like you would any brain criticality, like applied brain criticality paper, in order to enable the comparisons more easily. But I agree that in general, I would like to have more grounded ways of extracting this information, because it seems that it's important, it's quite relevant whether there is oscillatory activity that emerges above the background or not. I guess the standard answer would be that the fluctuations that we observe in the absence of those peaks. So if I would bandpass filter at some frequency that is kind of in the middle, in between those peaks, I would still be capturing the local fluctuations in synchronization of the neural populations. But of course, it's kind of a hypothesis.
Thank you.
Thank you.
Thank you. So now we move to the second reviewer, Gustavo Didier. Can you hear me?
Can you hear me?
Yes, perfect.
All right, thank you. Hi, Merlin, how are you? It's nice to meet you quasi in person.
Yes, that's nice. It's nice to talk to you.
Okay, so just wanted to say, I think that was a very beautiful presentation, matching a very beautiful thesis. So congratulations on all your work. It's very impressive. So I have actually two basic questions. One is very, very elementary. So to the best of my understanding, when we look at scale invariance in neuroscientific data, where we restrict ourselves to a narrow band, right? So it's typically low frequencies. You're talking about like something like between 1 Hertz and 8 Hertz, if I'm not mistaken, here. Okay, so but however, on the other hand, multifractal analysis is about very high frequency data, right? So can you please just briefly explain how we can reconcile these two things? Because in principle, one thing's band limited, then the other one actually goes down to very, very, very high frequencies. How can we reconcile these two things?
Well, firstly, for multiple analysis, what we're doing is we're estimating the scaling of some statistical quantity over a range of temporal scales. So the only condition that we have is that the data itself is scale invariant over that range of temporal scales, but it doesn't have to be high frequency. It could also be low frequency. So in the case of the low frequency brain fluctuations, the low frequency fluctuations of brain activity, like you can see here on this plot, as long as it's scale invariance, as it is in this case, we can actually apply the analysis and it will be meaningful. Of course, the interpretation then, well, it depends on what your framework is, and that has been historically one of the limiting factors of the multifactorial analysis literature in neuroscience, is that the interpretation was limited by the fact that we apply over these temporal scales the analysis methods, but then the interpretation of the results didn't necessarily follow from just carrying out the analysis. And then from the sort of neuroscience data, there's like different things that we might look at, but typically for the brain criticality, we're interested in oscillations, which appear as peaks, pronounced peaks in the power spectrum. And so typically we would, yes, narrowband filter the data, but the unique property of those oscillations is that if you look at the power spectrum of the envelope of those oscillations, so of the narrowband filter data, if you look at the sort of envelope of the narrowband filter data, it also has like a scale invariant power spectrum. So we can also carry out the analysis on that. So here, for instance, this is the alpha band, this power spectrum of the alpha band envelope that is coming from right over here.
I see.
Right over here, yeah.
I see. No, thank you. And my second and last question is a very broad question. So of course I don't expect like a full answer. Okay, so you even mentioned, like, for example, this author from Indiana, Beggs, right? He has a very interesting book on the brain criticality hypothesis. So how do you assess? It's not about his book, but it's about you. I mean, can you tell me a little bit? I mean, because in his book he's I understand he tries to connect brain dynamics, basic models of brain dynamics, if I remember correctly, branching processes, to the idea of brain criticality, right? And in particular about the optimal transport of information across the neural network, right? The brain neural network. So do you have any measure of understanding of how, of how we could connect in principle at least? I mean, these ideas about the transporting information optimally and so on to multifractality. I understand criticality and power laws, but multifractality, which is of course a more sophisticated notion of scale invariance. But it's not about his book, of course, about your general view of this.
So thank you for this very interesting question. I have also wondered that myself, actually, and how could we write sort of an extension of the branching process models for the brain that would include multifractality. So actually, the one discovery I made while writing the introduction to the thesis was that if you look at the sort of continuum limits of the branching process, so like a branching process where you sort of make the spatial resolution finer and finer, you end up with the same sort of equation that is in the Landau-Ginzburg model, at least for the excitatory field, where you have like a diffusion term, you have a nonlinear like a self-activation term. And basically, I think there seems to be some kind of connection. The other element I would say is that the introductory paper of the Landau-Ginzburg theory for cortical dynamics clearly states that their aim was to sort of do that a little bit. And what they show is that when you do the avalanche dynamic analysis of this model, it also has the same scaling exponents as the mean field directed percolation model class, in which the branching process also falls. So this seems to be like a potential way. I have not dug any further, but I'm sure that someone motivated enough could draw a link from this kind of equation. Discrete equations can be sort of transformed into field equations.
Yes, absolutely.
Yeah.
Absolutely. No, very interesting. Okay, thank you.
Thank you. Okay.
So thank you. Now we move to the third online reviewer, Victor Gersa.
Yeah.
Here? Yes.
Yeah, I'm here. No worries. Yeah.
Thank you.
So First of all, Merlin, félicitations. This was a very, very nice presentation. And it has confirmed actually your didactic capacity that you have shown already in your thesis. I really appreciate your introduction that you have had in the written document, in which you're trying to connect actually a wide range of aspects such as statistics, such as modeling, such as criticality, cumulance, etc. So very nice, all the way to brain theory, by the way. So it was nice. I did appreciate this. Let me get started actually on this level. When you were talking about brain theory, you mentioned this in the papers, but also in the background. You were making reference to criticality as a brain theory, and then you made the juxtaposition of self-organized criticality and criticality, made references to the self-organized criticality. You referenced Anna's paper, the PLOS computational biology paper, but there you stopped. Then you didn't go any further. So I was looking actually, can you make any statement about with the approaches that you have applied here, when applied to simulated or empirical data, making distinctions and statements distinguishing criticality versus self-organized criticality in the data? What are your thoughts on this?
Okay, well, thanks for the very interesting question. So the self-organized criticality concept is very powerful because it states that you can take a system where you don't have to tune anything and it will over time become critical. So I guess I have not dug too deep into the framework of self-organized criticality. Part of the reason why is I'm not sure how relevant it is with regards to like actually how the brain operates, because it appears that there is some kind of like it's not fully self-tuned. So there would be some kind of fluctuation dependent on homeostatic regulation and not like purely dependent on the dynamics of the system itself. So like a self-organized criticality system, necessarily the tuning of the system comes from its own dynamics. But in the case of the brain, that might be also coming from external factors. So with regards to actually measuring the data from the brain, I don't think I have gone far enough yet to sort of properly do that. What I would say is that possibly, I have no idea, but possibly multi-factor analysis could help tell the difference in experimental data between self-organized and the other kind of critical dynamics. So there is one mention I would say is that this model I have been working with in the first manuscript, it is a model that was intended to replicate self-organized bistable dynamics. So with the first order phase transition in the excitatory activity field, and with the inhibitory activity field, which basically sort of tunes that up to some constant. So there is like a constant here, psi, where the field R replenishes to. So that is a parameter that you can play with as well. That means it's not completely self-organized, but it's what they call quasi-self-organized. So I guess in that scenario, I'm more interested in quasi-self-organization, where you have one parameter that still controls the overall level of activity of the model, but the dynamics, there is still this nice timescale separation with the fast dynamics affecting like the slow dynamics over time, building a critical system. But I would say I have no idea how exactly this means that we get multi-factality then in the simulations or even in the brain. I'm not able to draw this kind of analytical link between the concept of self-organized criticality or bistability. But it's something I would be interested to investigate, of course.
Thank you, Merlin. It's actually my question was more from the data analysis perspective, because there are certain properties. But essentially, what you're pointing at is where one would have to make a mechanistic hypothesis in order to be able to address that. Yeah, and actually, let's stick to the question of mechanisms. In the beginning, during your presentation, I wrote it down actually. You said explicitly, and that was one of the subtitles, what does it mean that the brain is doing? Yeah, these were your words. Yeah, so based on the link between multifractality, yeah, and to the underlying brain processes that you're referring to, yeah, what is it you can actually conclude from this with regard to processing relevant to brain function?
So it's actually quite limited so far. And the reason is, well, we haven't been able to build like the necessary bridge between the two domains. Still, there is a few things that we can say. The first one is that since we observe multifractality in the brain, it means that there is like that restrains the domain of possible explanatory mechanisms for the brain activity. So it means that if we're looking for explaining how the brain's activity is organized, and we have a model that doesn't feature multifractality, it's probably incomplete because the brain data itself will display the multifractality. So that's maybe the most important points. But then apart from that, yeah, there's still quite little that we can conclude.
Mm-hmm. I was actually, the word incomplete you used earlier also in response to Anna's first question, when she pushed you into the direction monofractal versus multifractal. Yeah. And then when you take a monofractal, like a fluctuation analysis, you take a data analytic approach, you have a multifractal organization. Now you're looking for one particular scale, and you said it's incomplete. Yeah. I'll give you an example. Imagine you have a multimodal, a bimodal distribution, yeah, and you're taking a standard variational inference approach where you're approximating this with a Gaussian parameter distribution, which means if the two peaks are at -1 and +1 with your Gaussian approach, you will get 0 out. This is not incomplete. This is simply wrong. Yeah, is the approach, I'm rephrasing the question, is the information that you obtain incomplete, or is it potentially also wrong?
It's incomplete, but it can then lead to the wrong kind of conclusions. So it's only incomplete in the sense that what you get from only applying DFA still is a valid information on the power spectrum. On the multifractal spectrum. Mm-hmm. So applying DFA in this case here would give you the same C1 exponents that multifractalisis would do. So you would still get the information of where the mode of the spectrum is, but you would not know anything else about the multifractal spectrum. So in some cases where, for instance, there is joint variation of C1 and C2, that fluctuation of the mode of the time series might be due to something that's also related to other aspects of the multifractal spectrum. For instance, the mode and the width of the spectrum might vary together and in different ways depending on certain points in the parameter space of the system. So in that case, you could be misled by the appearance of only having one exponent, but still it's not wrong because you still get a valid measure of the mode of the multifractal spectrum.
Okay, thank you. I'm actually comfortable with this. Yeah. So great. If I may, technical question. Yeah. When you were talking about in your first paper, which I appreciated, neural field approaches, Wilson-Cowan. Then you went from the Wilson-Cowan classic representation to the field representation using the Ginzburg-Landau, the diffusion operator, yeah, as an approximation of the nearest neighbor interactions. In this formulation, you went then through the different parameterizations. My understanding is numerically, you did this with you implemented it on GPU in order to do proper parameter sweeps, and you used a Runge-Kutta procedure. I don't remember anymore what high order, third, fourth, sixth order, something like this, in order to run the simulations. But you focused particularly on the areas in the parameter spaces you had to, also sampling those that are close to the transitions, yeah? So the critical areas. In this area, the differential equations become stiff for the numerical algorithms for the simulations. How did you deal with this?
Well, there's multiple things. The first one is that actually in this case, it's kind of like the unique property of this model is the limiting factor mostly is the fact that there is this demographic noise, which is this square root, scales as a square root of the activity here. This term here means that we have to sample, when we do the sampling of the what's it called, the Fokker-Planck equation, we have to draw a Poisson distribution. We have to draw from a Poisson distribution. Actually, that's the limiting factor for the speed of the simulation is drawing a discrete random variable.
Sorry, did you say Fokker-Planck equation?
Yes.
So you simulated the Fokker-Planck version of it?
So in the
Sorry.
Sorry, there was a chime for the people who have to take a shuttle.
Okay. Yeah.
So yeah, in the way that the model is simulated, which I picked up from the people who first implemented this equation. So at the bottom of the slide, you separate the nonlinear dynamics from the multiplicative noise via the operator splitting. And actually that means that we don't have to go so high order on the Runge-Kutta approximation, because anyway, the operator splitting means that the error grows with delta t squared, which is our sort of simulation resolution. So we simulate the nonlinear dynamics using the Fokker-Planck equation, along with, we simulate the demographic noise using the Fokker-Planck equation, and sampling along that requires drawing from a Poisson distribution at some point. I don't really exactly remember where, but I know that it's the limiting factor. So like the rest of the simulation doesn't really matter, because the bottleneck will be the drawing, like sampling this demographic noise, in my experience at least.
Okay, but did you monitor the stiffness or the stability of this solution?
Yeah, the stability. Yeah, there is a divergence that emerges at some point, and you have to, but it's not like a real divergence. It's just from the way the equations are written. So it's simple to avoid it by just checking when it occurs and rewriting the equations appropriately. So yeah, I did do that work.
Thank you. Let me stop here. So thank you very much, Merlin.
Thank you.
Thank you. So now I think we will move to examiners in the room. What should we do with the microphone?
Yeah, that's a good question. Yeah.
Okay, so Laurie will talk now, and so
Sorry, can you please? We are
Okay,
sitting here like ducks. What is that
so
mean?
yeah, that's a good question. So there's like a few things I would like to mention in regards to that. So the first one is that, well, the simple fact is if you grow cortical slices in like a petri dish, actually they will also display critical avalanches, even though they have not been exposed to any kind of a natural stimuli, which might be scale invariant. So it's cortical neurons, they by themselves act in a critical manner. Then there is the other thing that when we put people in the image scanner, there is not actually so much well-structured stimuli that could have the form of a scale invariant process. So then it would be possible that something else not coming from the cortex could also be potentially stimulating it in a way that makes it scale invariant. Of course, it's possible, but I would say it doesn't appear very likely to me because the way we understand, at least the way, for instance, Beggs writes in his book about brain criticality is that it's if you think that the criticality is a necessary property of the brain, then it's natural that the brain will tune itself close to this critical point. So, for instance, placing someone in a dark imaging room will make it so that at first it might be affecting the person, but then the homeostatic regulation means that over time they will come back to the natural sort of close to criticality state, kind of no matter what we do. Even also that should in theory also hold for stimulus, different kinds of stimulus. We should become sort of used to them, and the brain should still return close to its homeostatically sort of convenient critical points in order to avoid being overwhelmed by natural stimuli that are like very intense. So I think what you described, that sort of SNR will affect the estimates, is quite likely, in fact. So if that's kind of one of my hypotheses for why we see this gradient in the first place would be that, well, we know that we have better SNR in towards more the occipital and parietal regions, and in the frontal regions especially, it can be that some subjects simply do not have very good SNR at all. So to me, that would be an indication that, well, it could be that there is less multifactoriality there, or simply that we cannot pick up the signal well enough. And I think it's also quite possible that in the gamma band, as you mentioned, we're just not capturing that at all. So I would be curious to see what, for instance, OPMs would achieve, or different recording modalities. And yeah, I think we should need some way to be able to measure like the dependence of SNR and multiplicity. It's true, I haven't investigated that, but it would be good to see if there is some kind of correlation pattern there as well.
There is quite often a correlation between the C1 and C2 cumulants in the cortical particles. So these also be something that is actually driven by the SNR in those particles, because then if you do have these oscillations which are showing the field with properties of multilaterality, so then you, I mean, do have both cumulants. But then if you have particles which show very little signal, so then it's likely that you cannot extract.
Yeah, I think that's a very reasonable explanation for what we observe in our results. So here, for instance, we see that I think what the correlation to the first principle component actually also probably measures some form of capturing the signal coming from the brain. And so it's quite likely that this plot here describes what you just mentioned, that we have some form of when there is no signal, there is no correlation, there is no information. I guess I'm not quite sure how to sort of disentangle that, but I guess my idea was that we could try to sort of first look for a better way to separate the different multifractal processes to see like what is their power level, like how big is our SNR, and then what the estimates looks like, so that we could have like remove this question of the correlation of C1 and C2 and focus on the actual scaling invariant processes themselves. Yeah, of course that would be like future works.
Yeah. So I'm I think on the same notions of that high loading on the first principle component is
Yes.
connected to the so maybe still about the first study. So the filter and fault level analysis yielded somewhat similar results. Would there even be any, or no, maybe that goes back to the general question of signal mixing, which you also alluded to in the presentation and previous answers. Could it be that, or were you expecting to have somehow higher grained results for the substrate analysis, because some of the mixing may not be there?
So when we do the parcel wise averaging, we're always hoping that what we average on the parcel is meaningful. I guess the problem here is that it might not be as straightforward as doing just that. So my hope nowadays is more that we would separate the sources in the sensor space and then project back individual maps from each process to the cortical surface, because it might be that we're projecting, like localizing sources currently, but we might also be adding mixing in between those sources unnecessarily before doing the analysis. So for instance, when I did the principal component analysis, I was a little bit hoping to find that I could separate the different components that way, but it doesn't work. Well, the components that we obtained, so the first principal component, I cannot estimate its scaling properties because it's not the proper scaling variant time series. So I'm kind of limited in that way there.
Yeah. Okay, thank you. Well,
thank you, Louis. Now I think I will ask questions myself before finishing with your supervisors. So I am not a neuroscientist, and maybe some of my questions will be naive. So, okay, first I found that you did a very good work. I could learn many things. I don't think it's I'm a signal processing person, so it's multifractality. It's not an easy topic. So I think you have a very good expertise. Quite impressive to have expertise in both sides, in neuroscience and signal processing. So regarding the chapter which is about the signal processing paper.
Yes. Ah, yes.
Last question, so you use you deal with impulsive noise, so why not something else?
So, well, it's because we're trying to model a phenomenon that we observed in our data, which is that, well, there are some noise impulses. And well, it was looking at, so at our results. So the multifactorial formalism says that the C2 exponent should be negative because it means that then the multifactorial spectrum is concave. And that's what you expect from like a normal time series. So we found empirically that there are many time series sometimes that have this very positive C2 values, which means that the analysis is not being carried out, like it doesn't make sense, basically, the end result we get. And looking at it, we found that in many of those cases, is this because there's some kind of noise impulse somewhere that completely breaks the computation of the statistics of the multi-resolution quantities. So actually, we tried a few different approaches. So we tried replacing the robust cumulants, natural cumulative estimators. We tried to replacing it with robust estimators. So there's the M estimators for the mean, and then we also use the QN for the scale. But actually, it didn't work because it didn't discard the same information across all the temporal scales. Because we compute those cumulants for each temporal scale separately, they discarded different information at different temporal scales.
Possible to do this and then to try to process this result at different scales.
Because that's really computing the cumulants is the last step before we do the linear regression that gives us the exponent. So it's really like at the end of the line, and it's already too late because we need to propagate the information between the temporal scales of what is a noise and what is an outlier and what isn't. So in this case, for instance, here, if you were to compute the robust cumulants for something like this, it would discard different proportion of the signal at different temporal scales, and that would mess up the scaling of the multi-resolution of this scaling function here of the C1 and C2 of J, and we wouldn't get proper estimates. We tried it, but it didn't exactly work.
So another question, probably the main question to me. So you work with MEG measurements. So each measurement is located at some spatial position. And so what I understand is that you do independent processing of all these time series. And so one could imagine that the parameters of your multifactorial process will be correlated spatially. So how could you promote that? Could you imagine some joint processing of time series?
Yeah, that's what we're hoping to do in the future is to sort of better separate the different processes. So the current pipeline for processing the data assumes a little bit that the parcel-wise time series are more or less independent from each other, or independent enough that it still makes sense. Because what we do is we apply a linear filter from the sensor space data to project it to the cortical surface. But of course, it doesn't add any information. So if there is correlation in the sensors that is not properly addressed by the linear filter, then there is nothing we can do about it. And it might also be that actually our linear filter approach is also not correct with regards to the kind of processes that we have dealing with, especially with scale-free processes. So there is a lot I think there's a lot to improve. But we wanted to start where the literature is currently as a basis for comparing, and then sort of push towards more advanced processing techniques. Because the other thing is that those more advanced processing techniques, they come also with some drawbacks in terms of interpretability of the results, or like having to tune some parameters, which might be quite tricky sometimes.
I have a question regarding the software. You mentioned open software.
Yes.
So I didn't understand if the software is targeted towards neuroscience, or is it only, and is it a generic software on multifactorial analysis? And how were there other softwares existing before the PhD?
Yes. Okay. So yeah, it's a general software for performing multiple analysis. So the aim was that we have this nice tool, and it would make sense to be able to use it in different scenarios. So the idea in the future is to interface it with different libraries for neuroscience data processing. But part of the challenge currently is that, well, as I mentioned, we don't have exactly like proper tools yet for extracting the individual scalar invariance processes. So the analysis that we're carrying out, the interpretation is still tricky to do. And the second thing is that there is some parameters to tune. So for instance, you need to integrate the time series if it's too irregular for the multi-factor analysis to be carried out. And it's still something that requires some degree of explaining. So for now, I guess this is more like destined towards people who are familiar with the concept of multi-factor analysis, but we're of course aiming to make it more available in the future to like wider neuroscience audience.
So it could be used for completely different applications.
Yes. Yeah, like anything that requires. Well, so okay, so you asked if there is also any other options. So when I started my PhD, I found this was a toolbox that was ported from a MATLAB toolbox, but it was quite slow at this point still, because here we're dealing. So for instance, in dealing with the image data, I had to analyze hundreds of thousands of time series, including the surrogates, and that would have taken forever with the currently available tools. So I adapted a Python toolbox to make it a bit faster.
Okay, so the fast is the important word.
Yeah, fast here is an important word. Yeah, because it's using the pi-wavelet implementation of wavelet analysis, so it's written in C, and it's very fast.
If you produce data as well, or just
With regards to
yes, collect data, make them available.
no.
Okay, okay, so I think I'm done, and now I will leave the floor to yes. Okay, again. So thank you, Merlin, for that presentation again. So as usual, you are doing a help. Although it has been a very intriguing and pleasurable process in the providing Europe and then collaborating with you. So thank you, both of you. I want to kind of play a utilitarian game for a moment. So imagine a clinician who gets your tool and says that, okay, this tool operational and scaling value. So first of all, the first thought would be that, okay, what is the fraction of variance of scaling variance that you can explain with C2 in version C1?
So, well, it's a great question. So what the C2 measures, so if you have a scalar invariant time series, it doesn't explain the variance, it explains the fluctuations in the variance of the signal over time. So basically the variance of the signal is entirely captured by the C1 metric, basically. So what C2 is dealing with is more like the temporal dynamics of the signal itself, rather than like a global metric that would be like variance of the signal.
Okay, so you would take by the definition that
okay,
kind of possible not to see one that makes it complementary information.
So it's complementary. It's information about things that happen locally, like how much local variation you're gonna have in your signal. So like you can, if you have a variance metric, it's a global information on your signal. So it's only informing you about the signal in general, but the C2 metric will tell you how much variation you can expect from one time point or one time window to another. So I think that in that sense, it's a useful metric.
Okay, very good. So
So now our hypothetical clinician has heard that imaging can be very useful for biomarkers. So he's very important clinician and he's very critical environment. So what would be your proposition to him to sell C2 as a new biomarker?
It would be pretty hard sell today. I think we still have a little of work to do on the tools for the analysis, but the gist of it would be that would be like a more complete picture. So ideally, well, I would say it's already pretty hard to sell today criticality analysis tools to clinicians as is. So adding on top of that multifactoriality it's going to be intriguing, but ideally it would explain the nature of the phase transition that you're dealing with, for instance, better than just looking at a single brain criticality metric that would be like DFA alpha or just a C1. Of course, today it's very hard to make this kind of prediction with confidence. So I probably would be pretty hard to sell this to a clinician today, but I could argue that it's important to develop those tools, and especially like for all parties involved. Like I think that the physicists, the signal processing people, they have these problems with the multiple analysis of the brain data that are complex problems that should motivate some developments of new efforts in those fields for like orthogonalizing multivector processes is one of them. We don't have established methods for doing it yet, but it would be a motivating example for those people. Also deriving analytical values for the C2 exponents from critical systems, from the equations deriving the analytical exponents. That's not something that's being done today. It would be also interesting to see more of that happening. And then after that, maybe we can bring it to the biomarkers, to the list of biomarkers that we sell to the clinicians.
Yes. So then for biomarkers, the statistical robustness is important. So let's say that your clinician knows that he needs to record X minutes of data to achieve a C1 estimate that has a maximum of 10% of product. How does that translate to C2?
Okay, well, great question. I think C2. So the drawback is that it's very sensitive to noise. So it's not going to give you a reliable estimate if there's a problem. But of course, the upside is that it's very much more able to detect when there is a problem, because it's very quickly going to be taking completely wrong values. So I think it's a very sensitive detector of anomalies in scale invariant time series. And for that, it can be immediately useful. Yes. So for instance, if you have like a large MEG data set and you run a DFA analysis on it, you wouldn't necessarily pick up like that there is outliers in some parts of your data, or that there's some serious problem in certain recording at a certain point in time. But multiple analysis most likely would be much more sensitive at picking that up and telling there is a problem with that time series at that time point. So in that sense, it could be used. Yeah.
Okay. And then as your discussion with the clinician is normal, front conclusion, if you want to know, but what then would be the physiological mechanism or other physiological mechanism that C2 would have?
Well, so in the case of the model that we have studied, it's a model for first observed phase transition. So that would be if. So that's a if. But if the part of the model that models the type of phase transition affects the multifactoriality that we estimate, you could say that this is going to help you detect a change, not in the critical point necessarily, but in the nature of the phase transition in the system, which might go from a gentle phase transition to something that might have like brutal bistability, for instance, in the case of epileptic seizure. So it could possibly be used. Of course, I cannot like 100% guarantee it from just simulation data, but that's an avenue where I think it's most likely to be useful.
Okay, good. Thank you. Okay, so I will complete the cycles around of PhD members. So first, Merlin, I would like to thank you for the quality of your presentation, of your work, your dedication throughout this 4 years and a half, and also the quality of your presentation today, which has been loving totally. Of course, that's been a challenging task to complete this PhD in a under the joint supervision of Matthias and myself, because this was the first collaboration between our two labs and with different cultures, expertise, and habits. Not everything has been easy, but to me, you really demonstrated great adaptability to the Finnish system, culture, through your determination. Second, I also believe that throughout your defense and your response to the numerous questions, so you have demonstrated a significant scientific maturity. Of course, this is because you process internally critical mindsets, but to me, and this is also because you have developed this ability to step back and reflect on your results, findings, and so on, particularly during this 4th year, and which was mandatory to obtain the PhD degree in Finland and eventually the dual degree. And to me, this is also quite rare in France, because PhD program in France typically lasts 3 years due to the limitation of the funding, and this usually results in less scientifically mature work and discussion. And I'm also convinced that your interdisciplinary contribution will open the new avenues for these adoptions of these signal processing tools in neuroscience, given the toolbox that you have developed so far in GFAS, but potentially also because of the expected impacts of your papers. And I'll get back to that point just in a minute. So before that, I would like to thank again Matthias for welcoming you into the critical oscillation lab in Helsinki. Matthias, but Satu as well, and of course, Shane for his strong support, facilitating your integration in the team, guiding you in writing grant applications, because it's quite usual in the Finnish system, that allows you to stay 2 years and a half in Finland, which was not expected at the beginning. Originally, this was only 1 year now. So thank you all for this. And so, of course, so I would like to congratulate you for all this contribution. But now I would like to ask you a couple of questions. That's originally, I didn't want to ask questions, but I'll contribute myself. Given the discussion, so that emerged with the reviewers, and also given the past results we obtained when we originally worked on energy data and structural analysis of such data with Virginie van Wassenaar, we somehow picked up that multifractality could reflect in terms of brain mechanisms, would be the hallmark of temporal multiplexing of cognitive processes, in the light of what is currently, what exists currently in regards to brain oscillations. You know, we have this cross-frequency coupling mechanism with, for instance, the phase of the lower oscillations modulating the amplitude of the faster one. So, and in the field of scale invariance, combining these different activities, different regimes of multifactoriality of irregularity somehow. Do you think that this could be now demonstrated with the tools that you have developed and with the connections with the models like the one you worked on?
Yeah, well, thanks, Philip, for the very kind review of the thesis work. But also, now to the question. I think it's possible now to formulate a bit differently the hypothesis for where we would find multifactoriality and what it would mean in brain data. So I would say, first of all, that the analysis that we did in this project that you mentioned was looking at the low frequency fluctuations of the recordings. And for that, we haven't really improved our understanding of where the multifactoriality could be coming from. So first step would be, I guess, to dig in that direction. But we wouldn't be able to, for that data, we wouldn't be able to rely on the brain criticality hypothesis because it does not explain that at all. And there is also issues, as Laurie mentioned, about the fact that the noise levels at the very low frequencies becomes like dominant. But then we could formulate some hypothesis on the nature of the multifactoriality in the oscillations. And I guess that would be a natural next step on the neuroscience side would be to ask the kind of question like, what kind of cognitive processes would generate multifactoriality? Like, is there even a difference between resting state multifactoriality in the oscillation dynamics and in the resting state and during task trial? One of the problems is that designing a task necessarily necessitates the subject to, well, usually at least, necessitates the subject to take some actions, and that will induce some kind of rhythm in the data. So the scale time experiments that you mentioned also tries to address this, but it still has the likelihood that the experiment design could also potentially induce the multifactoriality that we observe. So we need to have a good way of separating. This goes back to, again, we need a good way to separate the different multifactorial processes to determine which ones are cognitive from which ones are purely like sensory motor processes. And that will be very important because when you're designing this kind of experiment, as long as you cannot isolate the multifactorial process that corresponds to a cognitive process, then you cannot say decisively anything about the multifactoriality of the cognition.
Okay, so this was a question on the cognitive side, cognitive neuroscience side. So maybe moving to the clinical side would give us more possibilities. So you mentioned potentially the epilepsy as a target disorder to showcase the interest of new biomarkers related to multifactoriality. And so there is a lot of competitors, including Victor with the virtual brain and so on, who has developed a very nice digital twins to mimic mechanism that generates such kind of arrhythmic and seizures in the brain. And we also know that from Sheng's work that can derive in biomarkers related to stability or criticality and so on, that sometimes at the border of criticality and also moving to supercritical. So I'm not sure whether in that case we would see very significant multifactoriality. So I wonder whether we should look at pathology in the brain, where, for instance, we can demonstrate that the source of the anomaly in the brain dynamics is related to an unbalanced excitatory inhibitory ratio, like in schizophrenia or in spectral autism, for instance.
So I'm not sure how that would work, because the way the functional excitation inhibition ratio is calculated, as far as I understand, is based on DFA exclusively. I mean, it doesn't use multifactoriality in its definition, So we would maybe need to introduce new concepts, but that will require us first to clarify what is the link between even criticality and multifactoriality. Like, is multifactoriality like a byproduct? Is it like a essential part? I think it's still too far to say exactly how we could build multifactorial analysis as a biomarker. I would say it would be more, in my mind, it's more useful as for instance, a tool to design MF-based ICA that would separate multifactorial components, which would then, on which we would be able to apply the usual analysis. But it's hard to imagine multifactorial analysis as itself a tool for making biomarkers just yet. It feels not ready enough at this point.
Okay, thank you. And thank you, Philippe. Maybe someone who is a doctor wants to ask a question. It's now, or maybe online.
Yeah.
It's possible as well. Yes.
Very short question. I'm Thomas Baudel, doctor with a PhD at the and it's a very short question. So my background is not in neuroimaging or it's more in human-computer interaction and experimental psychology. In experimental psychology, there is this very intriguing concept, which is not very scientific, but it's evocative. It's that of the thickness of the present. Or sometimes some call it's the frequency clock of the human processor. It's about 10 Hertz. 10 Hertz is the, when there is a very pulsation, it goes from, when you go above 10 Hertz, it goes from a pulsation that you hear to a low sound. Same for the visual system, and same for the frequency system. My question is, in all your analysis, does this around 10 Hertz frequency appear as something that cadences the brain?
Okay, so first off, I need to mention that I'm not the best person to answer this kind of question in the room because there's Virginie. Oh, she's gone. Well, she was there. I guess she had to go. So the Virginie who works here in Neurospin, she is working exactly the kind of thing you mentioned. So first I'll say that, well, the closest thing in our analysis is the fact that, well, the alpha band is multifactorial. And actually part of a project that Philippe mentioned was trying to see if there is any kind of connection between the multifactoriality that we observe in the brain and the time perception. But it was inconclusive with the tools that we had at the time. So part of the idea would be, could we start another project on the data with the new tools that we have been developing? And that would be an interesting question. And then secondly, I recently revisited a paper from Virginie's lab, which investigated the time perception, and it clearly showed that this 10 Hertz rhythm affected the perception of the passage of time. So also there, that's confirming what you're talking about, but it's kind of separate from what I'm working on.
Other questions? No, I think we are done. So
maybe on Zoom, we need to make sure that no Finnish person wants to.
No, I see. It's okay. So do we stay online?